Hi all, in this video we're going to see about the structure of nephron along with its microscopic features. So this question has been asked as a diagram question in many university exams, and the microscopic structure also is important. So we'll quickly see about that. So first we should have a basic idea about the internal structure of the kidney. So we know the kidney has got outer cortex and a medulla, and it is covered by the renal capsule, and inside the medulla we've got these pyramidal structures which are called the renal pyramids. In fact, it is in the cortex and in the medulla that we've got the nephrons. The renal pyramids open into the papilla, which in turn drain into the minor calyx and the major calyx. This is the minor calyx and this bigger one is the major calyx, and they all drain into this area which is called the renal pelvis, which continues as the ureter. So we've got the cortex, which contains the nephrons, especially the glomeruli, PCT, DCT, and the cortical collecting ducts, and the medulla, which contains the loop of Henle, medullary collecting ducts, and the blood vessels. We'll be seeing the different parts of the nephron now, so don't worry about that. The renal pyramids, they are basically 8 to 12 conical masses which end at the papilla, then the minor calyx, major calyx, and the renal pelvis. So now we'll see the topic proper, which is the structure of a nephron. So the nephron begins with the renal corpuscle, which contains the Bowman's capsule, which contains this glomerulus, which is basically the blood vessel, the afferent and the efferent arteriole. After the Bowman's capsule, we've got the proximal convoluted region or the proximal convoluted tubule, and then we have the loop of Henle, which have got a thin descending limb and a thin ascending limb. Basically, the descending limb is actually thin only, but the ascending limb has got a thin ascending part as well as a thick ascending part. And it is in this thick ascending part that we've got the uh, juxtaglomerular apparatus. Then we've got the distal convoluted tubule or the distal tubule, which then joins the collecting duct via the connecting tubule. So the small part here which connects the distal tubule to the collecting duct is called the connecting tubule. And then the collecting duct, we've got two parts, that is the cortical collecting duct, which is present in the cortex, and the medullary collecting duct, which is present towards the medulla of the kidney. And finally we've got the collecting duct. So this is the basic structure. In this, I think you might be familiar with that outstretched structure of glomerulus, but in fact if you can show this, that the thick ascending limb goes between the afferent and efferent artery, if you can show that concept, is good, because we know we've got the juxtaglomerular apparatus there. So the basic structure of the nephron, we've got around 1 to 2 million per kidney. The segments are the renal corpuscle, which is the glomerulus plus Bowman's capsule, proximal convoluted tubule, loop of Henle, the descending and the ascending limbs, the distal convoluted tubule, and the collecting duct. Now we'll see the microscopic structure. So the different parts of the nephron has got specific microscopic structure. Don't think that everything is the same. The different parts, based on their structure and function, they've got different characteristics. So we'll quickly see what these characteristics are and what they mean. So first we'll see about the proximal convoluted tubule. The length is around 15 millimeter, diameter is around 40 micrometer. The parts are, see, it's got a pars convoluta, which is the convoluted part, and it's got a straight part which is called the pars recta, which leads to the loop of Henle. So the histology is basically, see, this is how the cell in the proximal convoluted tubule look like. You can see that there is this apical microvilli, a brush border, which is there to increase the surface area because maximum absorption occurs in this proximal convoluted tubule. And you can see that there are basolateral infoldings and numerous basal mitochondria. Not only that, between the cells we've got tight junctions also. So the major function of this proximal convoluted tubule is reabsorption. That is why we've got this brush border epithelium. So brush border, basal infoldings, lot of mitochondria, tight junction. Next we'll see about the loop of Henle. So as we said, the loop of Henle starts from the proximal convoluted tubule. It's got a descending limb, an ascending limb, and a thick ascending limb. The length is around 14 to 26 millimeter. Its main function is filtrate reabsorption as well as urine concentration, that is counter current mechanism. So the thin and the thick part has got specific microscopic structures. We will see what they are. In the thin limb, basically the cells are more flat. It is flat epithelial cells with smooth membranes. It's got lesser mitochondria as you can see, and is mostly involved in passive transport. That is why you can see that there are not much basal infoldings. Mitochondria is very less. The cell is very flat. Whereas for the thick ones, the cell is more cuboidal. There are more mitochondria when compared to the thin part. And here the importance is the basal infoldings are more than the apical. And this is in contrast with our proximal convoluted tubule, because proximal convoluted tubule had the brush border. So here more than on the top, the basal part has got more infoldings, and this is mainly because, and they've got a lot of mitochondria because active transport of solutes are required. And they end at the distal convoluted tubule near the macula densa. That is why I said the thick ascending limb ends with the juxtaglomerular apparatus. That is why I told we can show the diagram like this, so that we can show that the thick ascending limb is passing through the two arterioles. Next, we'll move on to the distal convoluted tubule. The length is around 5 mm. Here also the cell is cuboidal, just like the loop of Henle. Fewer apical infoldings, extensive basolateral infoldings, many mitochondria. Here also the function is reabsorption of sodium and water via aldosterone ADH, and it is otherwise relatively impermeable to water. So here it is more or less like the thick loop of Henle, except that you can see that it is more involved in water reabsorption via aldosterone and ADH. Next, we've got the collecting duct. The length is around 20 millimeters. Again, we've got the different parts: the cortical, medullary, outer medullary, inner medullary. The functions are again sodium and water reabsorption via aldosterone and ADH. So here the histology is similar to that of a distal convoluted tubule, but the difference is that the infoldings are less, mitochondria is less. They are cuboidal cells, but the basal infoldings are less, mitochondria is also less when compared to the distal convoluted tubule. And we've got two types of cells in the collecting duct: principal cells and intercalated cells. Principal cells are concerned with water and sodium reabsorption, whereas intercalated cells in acid secretion and bicarbonate transport. So I think this question, if it comes for the exam, it would be mostly like draw a diagram of the nephron along with its microscopic structure. Sometimes it can come as a short essay also, asking you to explain the microscopic structure of each segment of the nephron. So then you can draw this diagram showing the different parts, label it properly, so that the examiner knows you know the basic parts as well as the microscopic structure. So I hope this video was useful for you. Thank you.